Hello students, this is Dr. Nisha Varade, Department of Zoology, Shankarlal Khandelwal College, Akola, and today I am here with yet another topic, that is life cycle of Plasmodium vivax. To start with, we will first see the systematic position of Plasmodium vivax. This animal belongs to phylum Protozoa, subphylum Sporozoa, class Telosporia, genus Plasmodium, species vivax. Plasmodium is an endoparasite and digenetic, that is, it completes its life cycle in two hosts. First, the primary host or definitive host. Female Anopheles mosquito is the primary host, the organism which contains sexual phase of the parasite and is regarded as definitive host. The second is the intermediate host or the secondary host. Human is the secondary host. Human contains asexual phase of the parasite and develops symptoms of disease due to the presence of parasite and is termed as secondary host. The Plasmodium vivax life cycle is complex, including more than 10 stages of cellular differentiation. The parasite invades at least 4 types of cells within 2 different hosts. It causes vivax type of malaria where the malarial symptoms reoccur at every 48 hours, that is 2 days. Life cycle of Plasmodium vivax is divided into 2 phases. The first phase is known as asexual phase or schizogone in man, and second is sexual phase or sporogony in female Anopheles mosquito. Now we will start the first phase, that is asexual phase or schizogony in man. The asexual phase of life cycle of Plasmodium vivax starts with the first step, that is inoculation. The process by which the infective stage of the parasite is injected into the body of human is called inoculation. The infective stage of Plasmodium is sporozoite. Innumerable sporozoites are present in the salivary gland of female Anopheles mosquito, which feeds on human blood. Now we will see the structure of sporozoite. Sporozoites represent the infective form of Plasmodium. Each sporozoite is small, slender, spindle-shaped, covered by elastic, firm pellicle having longitudinally arranged contractile microtubules. They measure about 11 to 12 micron in length and 0.5 to 1 micron in breadth. The microtubules help in the gliding movement to swim in thick viscous human blood. Its anterior end is recognized on the basis of presence of apical cap made up of 3 or more concentric rings. A pair of reservoirs called secretory organelles open into the apical cap. The secretion of these organelles help to dissolve and penetrate the required tissue, that is liver or RBC cell. There is one vascular nucleus containing nucleolus. The cytoplasm also contains energy cells providing mitochondria and several convoluted tubules with unknown function. There is one vascular nucleus containing nucleolus. The cytoplasm also contains energy providing one mitochondria and several convoluted tubules with unknown function. There is a micropyle in the pellicle which corresponds to cytostome of other protozoans. When an infected female Anopheles mosquito bites a healthy person, it injects thousands of sporozoites along with saliva into the bloodstream of human being. Inside liver and RBC, different forms of sporozoid cause infection. The asexual cycle or the asexual phase or the schizogony in human is completed in following phases. The first is known as liver schizogony and the second is known as erythrocytic schizogony. Schizogony is the process of asexual reproduction by which Plasmodium undergoes asexual multiplication in liver cell and RBCs of human being. It occurs in human liver cell and in RBC. In liver cell it is known as liver schizogony and in RBC it is known as erythrocytic schizogony. Liver schizogony It is so called because it occurs in the liver of human being. Liver schizogony is again divided into two steps. The first is pre-erythrocytic phase and the second is known as exoerythrocytic phase. Pre-erythrocytic phase, when the sporozoid enters the blood, it remains active for about half an hour and disappears from the blood circulation. Then it enters into the parenchymatous cell of liver with the help of the lytic enzymes from the apical cap. Sporozoids in liver cell grow in size and become spherical in shape called the schizons. The nucleus of schizont multiply asexually, that is multiple fission occurs and forms thousands of merozoids. These gives pressure to the wall of liver cell and liberate out in the form of cryptozoids or cryptomerozoids through ruptured liver cell. This process of formation of many cryptozoids from single sporozoid in liver cell is called pre-erythrocytic schizogony. The second is the exoerythrocytic schizogony or exoerythrocytic phase. The cryptozoids are ready to infect the fresh liver cell where they grow and become schizont. The same process is repeated several times. The liberated merozoids in this phase is called metacryptozoids. The process of formation of many metacryptozoids from the cryptozoids in liver cell is called the exoerythrocytic schizogony. Some metacryptozoids are smaller in size called micrometacryptozoids and some are larger in size called macrometacryptozoids. The micrometacryptozoids enter the RBCs to start the erythrocytic phase, while the macrometacryptozoids infect the fresh liver cells to continue exoerythrocytic phase. Thus, in both these phases, large number of liver cells are damaged and lead to the malfunctioning of liver. Between the initial sporozoid infection and the first appearance of the parasites in the blood is termed the pre-patent period. In Plasmodium vivax, the duration is 8 days. The period between infection of sporozoid and the appearance of first malarial symptom, that can be fever or chillness, is termed the incubation period, which is of 14 days duration in Plasmodium vivax. Erythrocytic schizogony. This cycle starts when the micrometacryptozoids enter the erythrocytes and they become rounded with large nucleus and grow in size by ingesting hemoglobin of RBC. This stage is known as trophozoid stage. Inside the trophozoid, a large non-contractile vacuole appears which pushes the nucleus towards periphery and forms a ring-like structure known as signet ring stage. Trophozoids enlarge and vacuole starts disappearing and develops pseudopodial processes in the cytoplasm and changed into amoeboid stage. This stage is called amoeboid stage. The amoeboid stage feeds completely the component of corpuscle in the form of hemoglobin. During feeding, the hemoglobin breaks down into hematin and globin. The globin is absorbed by the cell and hematin is deposited in the form of hemozoin. This hemozoin is the toxic malarial pigment which causes fever. The amoeboid trophozoids after feeding becomes rounded, grow in size and becomes erythrocytic schizont. Asexual multiplication takes place in schizont and forms merozoids which give pressure to the wall of weak RBC and liberate out in the form of erythrocytic merozoids. These merozoids are arranged towards the periphery of the periphery due to the presence of hemozoin at the center. The arrangement is just like the arrangement of petals in rose flower. So this stage is called as rosette stage. Numerous yellow eosinophilic granules appear in the cytoplasm of the host corpuscle, which are called Schaffner's granules. These dots are believed to be the antigen excreted by the parasites. The process of formation of merozoids in the RBCs from the metacryptozoid is called erythrocytic schizogony. It completes about 48 hours in human beings. many merozoites enter the fresh RBC and repeat the erythrocytic cycle. The last step in the asexual phase of life cycle of Plasmodium vivax in humans is the formation of gametocytes. After some generations of erythrocytic cycles, some of the merozoites invade the new RBC. They grow in size but do not develop into schizones. Instead, they develop into gametocytes. The gametocytes are of two types. The first are known as macrogametocyte and the second are known as microgametocytes. Macrogametocytes or female gametocytes are larger and spherical in shape. They have small compact peripheral nucleus. They have reserved food material and the cytoplasm is dark in color. The microgametocytes or the male gametocytes are smaller, motile and few in number. They have large centrally placed nuclei. They lack reserved food material, stains faintly, hence the cytoplasm is light in color and clear. Further development of gametocyte stops in man and only possible in mosquito due to the low body temperature of female Anopheles mosquito. With this, the asexual phase of life cycle of Plasmodium vivax ends here and sexual phase starts in the female Anopheles mosquito. We will continue this sexual phase of life cycle of Plasmodium vivax in our next lecture. Thank you very much.